All right, so open up your Conflict. You've got 2 lectures. This lecture is the first lecture.
Wait, 2 lectures today?
Oh my God. All right, just pay attention.
It's recording.
All right, you guys ready?
It's recording, so whenever you're ready to start.
All right, let me start now.
And now. Action. Because I didn't credit for it. I get credit.
All right, so step 1, take out some paper or your compost so you can write this down. And then if you look at everything that's up there, it says this is lecture number 13. I feel really bad because I'm only giving you 12 lectures in 15 weeks. That's not very many. So this unit is super easy. So if for whatever reason your grade isn't that great, let's say in the test category, it should be bumped up by this test, because it's on genetics, Punnett squares, things like that that you already know about. And then I've already given you the calendar. If for whatever reason you lost the calendar, there's more calendars up there at the front. And so basically you can just see what's going on. So that way you know when due dates are and when the final exam is and all that kind of stuff. And when your project is due, as I mentioned. You should have your project roughed out by now. If I don't, you better get it to me by tomorrow, all right? And on your desk, you have 1 page that I want you to do today. It's the front page. It's on heredity. It's basically Punnett squares. You have to do 1 through 14. And that's about it. And we'll watch the movie at the end. I was going to show you this movie right here, but I think I'm going to change my mind and show you more of the island. If you're good.
Yeah.
All right, so put your pens down and listen for a second. All right, so science projects that can change the world. So all you have to do is just listen. So you may or may not be aware of the current world population as of yesterday, about over 7 billion. And then in the United States, we're 322 million. So our percentage of 7 billion, not so big. World population, basically on the average, there's 1 birth every 8 seconds, and there's 1 death every 12 seconds. So I went to the U.S. Department of Commerce, and it's kind of cool because you can see the numbers continually to move statistically. And once again, this is taken yesterday. So approximately 3 billion people lack potable, which means drinking water, electricity, and health care. So out of that 7 billion, 3 billion don't have any of these things that we take for granted. So solution, potable water. The solution is called the Rainmakers. Basically what the Rainmaker is, it's a desalination unit, and what desalination means is you take the salt out of the water and make it fresh water. So a desalination unit is roughly the size of a flatbed truck that relies on the conventional power source to distill seawater into fresh water. So you can see that's solar powered right there. And the key thing that's really cool about this is basically stack a bunch of these on a barge, and the barge is movable, so you take it to areas where people actually need fresh water. So this is from last month, 2015. So basically generating electricity is really easy. You just need a source of heat. You get heat, that makes steam. Steam makes basically a turbine spin, and as it spins, it's going to generate electricity. That's like the basis for all electricity. There's coal, creates heat. This one right here is nuclear power, making heat to make steam, once again causing things to spin around. Generator generates electricity. So electricity, solution, bipower generator that turns 1 hour of writing into 24 hours of electricity. This is the prototype right here. So to create energy, users must pedal a recumbent stationary bike for 1 hour. It activates a flywheel. This in turn spins the generator and charges the battery. No pollution. So, for example, you get on the bike, do it for an hour, that gives you 24 hours of power to use, charge your cell phone or whatever it might be that you need. This is taken from this month, 2015. Electricity, this one right here, I don't know about it, but they're doing research on this as well. So, it could come from the first core, as you know, is very hot. And basically what graphene is really cool. It's through nanotechnology. It's 100 times stronger than steel, and it conducts basically thermal, which is heat. So this is showing what graphene is. So what they would like to do is get a graphene string, which will be used to create a cable, which in turn will be placed into the ground to bring the energy or conduct heat at the Earth's core and bring it to the surface. That'd be unlimited energy and it's pollution free. This is from last month, 2015. All right, so this guy right here, he's actually the one who's like sponsoring all of these projects. He came up with the 5-hour energy drink, and his net worth is $1.5 billion, so he's able to fund all these research topics, which are actually really cool. His background is high school graduate. It's a good goal for you. His next thing is he's actually only in college for 1 year, and then he dropped out. So after he dropped out, he became a monk for 12 years. So when you're a monk, you have a lot of time on your hands to think about things. So obviously he was thinking about these things to basically solve problems in the world. So that's actually really cool. So all these things are actually really simplistic devices to basically help out a lot of people. So I'm hoping with your science project that you'll have something really cool, really unique. It shouldn't be a rubber egg or anything like that. Like I said, I'm going to go over looking at your project topics over the break and give you some feedback and get it back to you in about a week. So like I said, most of you looks really good. There's only a couple ones that I bounced back to people that just, were like at an elementary level. All right, so here's my segue. Do you know who else was a monk? That's right, Gregor Mendel. This is the stuff you write down now. All right, so this is actually the first part of the book activity. This is the first part of the lecture. So as I mentioned, the lecture is really easy. Basically, it's just vocabulary, and the vocabulary is very easy as well, meaning that you don't have like big words like the living plasmic reticulum. You have very simple, easy words that are actually already part of your vocabulary. So genetics was founded with the work of Gregor Johann Mendel, and basically he was an Austrian monk, and he experimented with garden pea plants. So all of that you need to know, and that was actually, like I said, that was like one of the first questions in the book activity. All right, so like for every unit, I always give you at least one famous person. For the last unit, it was basically Louis Pasteur. We talked about pasteurization, and Francisco Redi, who proved or disproved spontaneous generation of life from decaying meat by doing a simple experiment. Let's get a simple experiment to prove things. All right, next slide. So genetics was founded with the work of Gregor Johann Mendel, Austrian monk. So on your test, I'll just give you some random names, and you pick the correct name. It's this guy right here. I might write Johann Mendel. That'd be someone different. All right, so this is how Mendel performed his experiments. It was very revolutionary. This is actually how he did it with self-pollination of pea plants. This seems more interesting. This is his actual garden, which is in existence today. So if you look at it, it's very simple. You've got dominant versus recessive. This is obviously dominant. This is recessive. That's how he came up with all these results of dominant versus recessive traits. Bless you.
Bless you. Bless you.
This part right here you don't need to write down. This gives you a timeline of what's going on. So for example, next unit and next semester is basically getting into evolution. This unit we kind of wrap up genetics and all that good stuff. So if you look at Mendel, once again, it's like in the 1800s when I talked about Francisco Redi, that was in the 1600s. And you can just see how things tend to evolve. All right, so these are the terms that I'd like you to know. Like I said, they're very easy. In fact, one of the questions in the book activity asks you what does the term inherit mean and what does heredity mean. So obviously you got the word heredity within the word inherit. So heredity is the transmission of characteristics from parents to offspring. So on your test, you're going to have a whole area of you match the vocabulary with the correct term. As I mentioned, all these terms are very generic. They're already in your lexicon or your vocabulary. Next one right here. Yeah, I like that word. SAT word. Next one is trait. So a trait is a specific characteristic, and once again, I just use obvious, traits and examples, so that way, if you understand that, then you can get into the big picture of other types of traits that might be a little more complex. Traits that could be polygenic, meaning that there's more than one allele that actually codes for the trait. So obvious traits would be, you can distinguish the difference between something tall and something short. That's what he did with the pea plants. And you can also distinguish different colors, like he did color of the flowers, and here you can see the color of the eyes. So once again, a trait is basically a specific characteristic, and the two examples I'm using are height and eye color. Last time when we talked about cell division, the specific example I kept using was skin for mitosis, because mitosis is for growth. So I'm going to just use these two because basically you look at height and color for the pea plants to come up with this theory. Yep.
So if like if the heredity and genes like code for like color of
Yep.
hair, how come on like some dogs you have like white fur and like other parts stuff?
Because you're going to find out that it could be like a recessive trait. So when I go through this, you're going to find out you have the parents, just like when you had in cell division, you have the parents give rise to daughter cells. In this case, you have parents that give rise to offspring, and that offspring can have a hidden trait that won't show up in that generation, but can show up in the second generation by probability. That's what a Punnett square is.
Okay, but in the same organism, it'll have two different traits showing.
Meaning like a dominant and a recessive.
Okay.
Well, no, that's because you can have multiple genes, can have multiple traits. So don't just think that one gene is specific for everything. Remember, you got 6 billion nucleotides, and out of those billions of nucleotides, they all do different things. So it's very specific.
Okay, so why in like one hair follicle the dark gene, the dominant gene will be like the phenotype, but in another hair follicle the white gene will be the phenotype?
So you say you have dark hair on your arms and white hair up here?
No, like with dogs.
That's basically, that's how they're bred. That's like, for example, if you have a red flower and a white flower, it breeds to a pink flower. You have combinations. That's what genetic engineering is. I know, I'm sorry to disappoint. All right, so the next one is molecular genetics. So that's actually what we talked about in the previous unit. And molecular genetics is the study of the structure and function of chromosomes and genes. So like I said, your next exam is easy. It's Mendelian genetics, but your next exam following that that's going to be on the final is going to be how DNA is replicated, and it's also going to cover protein synthesis, which is a little challenging. So you want to make sure you do really well in the next exam, obviously on the final. So looking at molecular genetics, this is what you did last time. You made a karyotype, and a karyotype is a picture of an individual's chromosomes. That was on the test. You put them into homologous pairs, going from tall to small. That was on the test. You've got 22 pairs of autosomes. That was on the test. And you have 1 pair of sex chromosomes. That was on the test. It was XX for female, XY for male. If you count up the total number of chromosomes, you have 46 chromosomes, or 23 pairs when you combine both autosomes and sex chromosomes. So that was the point of you doing a karyotype, so you could understand that whole concept right there. So for this unit, this is what we're focusing on right here, is the Punnett square and probability of outcome of genetic traits. So once again, you've got your DNA inside the nucleus, you pull it out, you've got chromosomes. With each chromosome, you have a gene, which is a segment of DNA. Yep. So you got 2 more exams. You've got the exam when you come back, and then you got the final. Yep, it'll be comprehensive, and I'll give you a study guide. But let's focus on this right now. Be in the present. That's why it's a gift. So Mendel observed 7 characteristics in pea plants. So basically, he looked at what are called contrasting traits. So contrasting traits, out of the 7, I just picked 3. Once again, I picked 3 that are obvious. They're all very obvious, but for example, I'm going to keep focusing on height because that's something that you can readily observe. So plant height, the stems are either going to be short or long, so that's one trait. The next trait is seed color. The seed would either be yellow or green, so that's a contrasting trait. And flower color, like I showed you in the very first picture when we looked at his pea plants, you could tell that purple was going to be dominant by doing these experiments, so purple or white. So the thing is, he looked at these traits individually, meaning he wasn't looking at plant height and flower color at the same time. He was specifically breeding for height, specifically breeding for seed color, and specifically breeding for flower color. on any other 4 traits that are right here in the picture. Yep.
If he wasn't a monk, would he have managed to do all of this?
Who knows? But actually, I used to teach a course at UCLA for the Summer Institute for the Gifted. And it's all about people that come up with creations or inventions and stuff like that. And a lot of stuff happened, like stuff that we have today in like the 1900s, when there really wasn't TV or anything like that. So a lot of people had a lot of time to focus on a lot of different things. Whereas now, holy cow, there's so much distraction, social media, TV, a billion channels, Netflix, all that stuff. So. All right, next slide. So plant height, seed color, once again you can see the flower color right here, purple versus white. You can see the height, tall versus short. Seed color, once again, yellow versus green. And then you had a couple of other things, but those are the 3 that I think will probably stick with you. Then showing you an enlargement of it, once again, just showing you contrasting traits. All right, so as I've said a couple of times, I'd like you to know that they were contrasting traits and you did them individually.
On the test, we have to know like yellow and green.
No. So when you're looking at a contrasting trait, you're only doing 1 trait at a time. So like I said, tall versus short. And like I said, it's the obvious ones that you'll see, like the color, purple versus white. And the way that you would know that they are dominant or recessive is based on his experiments. Alright, so I looked at 7 traits in pea plants, and we did each one individually. And I gave you 3 examples that are pretty easy to remember. Once again, just showing you figured out if it was dominant or recessive based on the offspring. Alright, so this was another one of the book questions that maybe 1 or 2 people had questions about. So basically we're talking about something that's pure in genetics. So we're going to go through all the steps of what he did. So first thing he did is he began by growing plants that were pure for each trait. So what that means is plants that are pure for a trait always produce offspring for that specific pure trait. And hopefully the picture below will help make that even more clear. So for example, if I have something that's 100% pure for red, and I breed it with something that's 100% pure for red, my offspring are always going to be 100% pure for that trait. So it's a purebred. So that's a dominant trait. So if I have something that's 100% white with something that's 100% white, and I breed those 2 together, the offspring are going to be 100% white. So those are called purebred or pure traits. So if I had bred something that was 100% red with something that's 100% white, you're not going to get a purebred, you're going to get a mixture. So that was his first concept, is basically he did stuff. Yep.
So like dogs?
Exactly, dogs. So dogs that are purebreds are very expensive because you're going through a certain lineage. Same thing when you're going to purchase a racehorse. You're going to get a purebred racehorse, and because you want that to come from a good stock and a good breed.
How do we know they were pure?
They do genetic testing and all that stuff.
In 1800?
Yeah, they had a time machine that came to the future and then back. That's my final answer. All right, next slide. So we began by growing plants that were pure for each trait. Plants that are pure for a trait always produce offspring for that specific pure trait. So 100% red was 100% red gives you 100% red. All right, so once again, this is just vocabulary. On your test, you'll have a section where you match up the vocabulary word with its definition. So, like I said, you've heard of the term strain before, but when you talk about genetics, the term strain denotes plants that are pure for a specific trait. So when we talked about cell division, we talked about parent cells giving rise to daughter cells. Well, you have almost the same type of terminology here. You have what are called the first strain, is referred to as the parental generation, or also referred to as P1. And then if you look at the picture right here, this is actually a picture of a pedigree that we'll talk about more next time. A pedigree is basically your lineage or your family tree. So obviously you start off with the parents, then you get the second generation, third generation, or F1, F2, and F3, and so on. So the term strain basically denotes plants that are pure for a specific trait. The first strain is referred to as the parental generation or the P1 generation. And once again, showing where all this stuff comes from. You've got the nucleus. Inside the nucleus are chromosomes. Take out a little bit of that, and there's the DNA. And a segment of DNA is the definition of what a gene is. And genes code for specific traits. So looking at this picture right here, so these are the parents right here. So you have a tall with a short plant. You breed them together. This is a recessive trait. It's actually going to be masked or hidden in the first generation or the F1 generation. You breed these two together, and this will be the next generation. So in this generation, the F2 generation, you can see where this trait is actually being seen now, which is the recessive trait. And once again, if you look at the plants and the numbers you use, I keep telling you it's a good thing to use like a large sample, and once again 1,000 is a good number. So he figured out that 787 plants were tall and 277 were short. So once again, that being dominant trait, short being the recessive trait for the P plants. All right, so when the plants matured, he recorded the number of each type of offspring produced by the P1 plant. So once again, that's the parents. So the next generation that comes from the parents is referred to as the F1 generation or the first filial generation. And then from there you can go to an F2 generation, and that's typically as far as we go. But you can go to F3 and so on and so on. So you look at these two seeds right here. So round is dominant, you've got two capital R's. Wrinkled is recessive, and once again the P stands for parent. So you breed these two seeds together and you get a mixture, but the dominant trait, big R, little r, is the one you're going to see. So you don't see the recessive trait because dominant basically dominates over the recessive trait. And then in the F1 generation, you breed big R, little r with big R, little r. And this is the outcome, and this is what you figure out in a Punnett square. So in the F2 generation, you now have 3 round and 1 wrinkled, thereby this being, once again, the dominant trait, and that one being the recessive trait. So that's basically how he did all his experiments with contrasting traits. Raise your hand if you need more time. All right. So parents, always start off with the parents, then you go to the F1 generation, then you go to the F2 generation. Like I said, you could just keep breeding and breeding.
Say you have two F1s and one is tall and short, are you still the same outcome?
Nope, because that's a different type of cross. That'd be big R, little r, and then you would breed them with 2 small r's.
So how did they obtain pure, how did they obtain pure AP1 if they're most of them is probably mixing?
Just by doing large numbers, and then you figure it out. And like I said, you did like 1 trait at a time. And you'll see when we did the Punnett square. That's a nice picture.
Do you want us writing that? Oh.
All right, so this part right here is more vocabulary. So you should definitely know what an allele is. So what allele, that's what we're talking about right now. It's referred to as an alternative form of a gene. So when you're doing your Punnett square, you're plugging in alleles. That's what the letters represent. So letters are used to represent alleles. So like I said, make believe like you don't know anything. And when you're doing Punnett squares, you got to know the vocabulary because these are basically word problems. So letters are used to represent alleles. And once again, in this case, we're going to look at height. So a dominant trait is always going to be in capital letters. In this case, that's tallness. Recessive trait is always going to be 2 small lowercase letters. And then putting that all together is you have a gene. So a gene is a segment of DNA. In fact, there's the picture. There's DNA. It says right there, gene. Once again, that's the chromosome. You unwind it, you got the double helix. So a gene is a segment of DNA that contains coding for a polypeptide or a protein. So remember, DNA says, I want this protein, and then RNA goes out and makes that protein. And a gene is also, another term, is called a unit of heredity information because it's passing on, once again, things that you inherit, specific traits. So here's the chromosome here, there's the chromosome there, and here's showing you an allele. So this is an allele, which is an alternative form of a gene. In this case, we're looking at the color of the flower. So purple versus white. So if I were to plug that into a Punnett square, I would make this like a capital P for purple because that's dominant, and then a small p for white because that's recessive, and then I'd see what the outcome is by putting it into a Punnett square, and you would see the probability of the outcome of what colors are going to show up with that type of a mix. So like I said on the test, there's going to be a bunch of vocabulary stuff that I want you to know. So if you don't know the vocabulary, you can't really solve the problems. And allele, alternative form of a gene, segment of DNA. The letters that you plug into a Punnett square are basically coding for alleles. And once again, they're always going to tell you, for example, that T stands for tallness. You can't like infer or imply that T is for tallness. They'll let you know that within the word problem. So I'm hoping most of this stuff is a review. Like I said, the vocabulary is very easy. But like I said, a lot of things sound like they mean the same thing, and they kind of do, but they're very specific to the words that are being used in these definitions.
Third organism, tall is always dominant, or is it for?
It depends. You can't say every or
So,
all.
I'm gonna make tests on a bunch of different things.
Any person.
Teachers on the
Oh, if you look at the family tree and just see, like for example, things that are inherited like male pattern baldness. That's something that.
So you have like a tall like component and a small component.
Yeah, so you breed them and see what happens. Excuse me. All right, next slide. So, this is what we're talking about. So when you think of Punnett square, this is what we're working with in this unit. So today's all about how to do a Punnett square and the vocabulary that goes into creating a Punnett square. And then the next lecture is basically talking about things that are actually Mendel's laws and things that are on, like for example, X-linked traits like color blindness and things like that. So when you think of a Punnett square, think of the P for probability or predicting. So Punnett square is used for predicting the probability that certain traits will be inherited by offspring. So there's 2 Punnett squares that I would like you to draw into your notes, so that way you'll understand when you look at the handout that I have in front of you, and any word problem that you see, you'll be able to do it. So the P for Punnett square is the P for probability. So basically what you do, this is a heterozygous cross, and then, because people keep asking, well, what happens if you do this and you do that? Well, you plug it into the Punnett square, that's how you figure out the probability. So the nomenclature and the way you do it is super easy. So this is the female right here, the ovule. This is the male right here, the pollen grain. So you plug in the letters, and you always do capital letters first. So for example, you put a big T over here, big T down. So that's tall. Big T over here, little t down. Still tall because that's dominant. big T always goes in front, then little t over, still tall, then little t over to here, and little t down right there, so you have short. So your outcome with this cross between this and this, you would have 3 tall and 1 short. Each box could be 25%, so 75% of the offspring would be tall, 25% would be short. So like I said, very easy stuff to plug that in. Yep, so I'd like you to draw this box. So if you look at that box up there, or that Punnett square, it's very similar to number 7 on the handout that I want you to do today. So number 7 says, complete the Punnett square to show the F2 generation. So instead of big T, little t, they use big W, little w. So like I said, try and do it on your own. Bring in the capital letters down first and the smaller lowercase letters next. So by having this one down, you should easily be able to do number 7 on the sheet in front of you, which talks about heredity. All right, so once again, it's all about probability. Plugging things into a Punnett square, so you should be able to do that on the test. So on the test, I'll give you scratch paper. Once again, it's super easy to do. So big T down, big T over, big T over, little t down, big T down, little t over, little t, 3 tall, 1 short. All right, so if you have this stuff in your notes, it's easy for you to refer to, and if you can also use that, obviously, to help you with the handout that's on your desk.
Are we slides over?
Don't know. Not enough, in my opinion.
It's a lot of writing, really only 4th page almost.
You're welcome. All right, raise your hand one more time. All right, so looking at the Punnett square, the next one is, once again, vocabulary. I'd like you to write this Punnett square down as well. So this one will be on your test for matching with the vocab part. So homozygous, when both alleles of a pair are alike or the same, the organism is said to be homozygous for that characteristic. So when you look at the letters, so for example, you have 2 capital letters there, and it's letting you know that that equals red. So if I were to have written this, I would have had 2 capital R's to equal red. But I Googled the picture, and then I actually made the PowerPoint that goes with the image that you're going to see. So that's homozygous because 2 capital letters are the same. So the opposite of that is going to be 2 lowercase letters. That's going to be for blue. So that's a homozygous cross. So they're going to ask you to do a homozygous cross and a heterozygous cross. So we just did a heterozygous cross on the previous slide. So on this slide right here is, once again, it's really easy to do, is you're looking at flamingos, and when you bring it down, capital letter always goes first. That's why I picked it, because it's pretty easy to see. Big A down, little a over, big A down, little a over, big A down, little a over, big A down, little a over. So the offspring here is going to be 100% red. So copy that Punnett square down as well. And then after you do that, look at number 6 on the handout on your desk. And basically, instead of having 2 large A's and 2 lowercase a's, you have 2 large W's and 2 lowercase W's. So like I said, very easy to do. Those are the only 2 possibilities that you can have.
You want us copying down the picture as well?
Yep. 'Cause
No, I
these
mean
are-
like the flamingo.
No, you don't have to put it apart unless you want to. It's an option. It's going to be optional today. Ladies. I just thought it was odd that they chose A for red.
I thought it'd be awkward, you know?
All right, raise your hand 2 more times. All right. So homozygous, when both alleles of a pair are alike or the same, the organism is said to be homozygous for that characteristic. So if they ask you to do a homozygous cross, you basically know how to do it because you have it in your notes, and you also have it on the sheet in front of you to practice. All right, so the opposite of the same is things that are different. So heterozygous, once again, the key is underlined for you, is different. So you would have big letter mixed with a smaller letter or lowercase letter. So we actually already did this one. So this would be a heterozygous cross. So you already have that in your notes. So you're able to do number 7 on the sheet in front of you. From the previous slide, you can do number 6. So heterozygous, when the two alleles in the pair are different, the organism is heterozygous for that characteristic. So actually, when I Googled the picture for pea plants, I thought it was kind of cool because these are actually the three colors, and I've never really seen that color before. And then, of course, this is really important right here. Give peas a chance. See that? Chance.
My doctor has those in her bathroom, or in like the bathroom office.
Aw.
In the bathroom.
So if you look at the pea plant colors right here, these are once again for the seed colors. So once again, if they're different, for example, big T, little t, once again, big A, little a. And once again, you're looking at seed color here. So you've got, bring the big A down, big A over, big A over, little a down, big A down, little a over, little a, little a. So you got 3 yellow versus 1 green. Or 75% of the seeds will be yellow and 25% will be green. So I've only assigned the first page in the packet today, and I'm not going to check it because we'll all check it, after the break. But there's also a page on the very back that's related to vocabulary. So these terms here will also help you within the vocabulary part of the packet.
Our test, and Monday will come back.
No? Should be a calendar on your desk. If not, there's one up there for you, so you can see when things are happening.
We're gonna check.
All right, so the next term is genotype. So genotype refers to the genetic makeup, and genotype actually has the word gene in it, so it's kind of hard for you to confuse it for anything else. So if I were to ask you the genotype for things, you would give me a bunch of letters or alleles. So there's only 3 possible combinations that you can give me. First would be big R, little r, 2 small r's, or 2 big r's. So this right here, if I asked you for the genotype of these flowers, you would not give me the color of the flowers because I'm not asking for that. You would give me the genotype, which would be the combination of alleles. So if I asked you for the genotype of red flower, you would say either 2 big R's or big R, little r. If I asked you the genotype for a white flower, you would say 2 small r's. Because once again, white's going to be recessive. So looking at the genotype, codes for what you're actually going to see, which is called the physical expression or the phenotype.
You have to remember if that red is
Nope.
on there.
Everything will be to you on the roll call. All right, raise your hand a few more times. So once again, really easy concept. The word gene is within the word genotype, and genes are represented by alleles, and alleles are represented by letters. All right, so, and don't be afraid here, people. It's got the word ratio. Every time people see ratio, they think of math, and they go, oh. But this is very easy. So this could be an exact test question right here. Just saying, this could be an exact test question right here. So if you cross big T, with big T, little t, and you've already done that in the Punnett square, and if I were to ask you the genotypic ratio, you should be able to figure it out. So you plug them into a Punnett square, and hey, let's play school. How many sets of big T's do you have here? One, very good. Good class. How many sets of big T, little t do you have? Very good, good class. How many of these do you have? One. That's the ratio, it's 1 to 2 to 1. The only reason I bring that up is because somebody was really confused by that last period. I'm like, huh? The other thing is when you see it on the test, you're not going to see these letters. I just put these letters here so you know what it indicates. You're going to see 1 to 2 to 1. And once again, you always do capital letters first or the dominant trait first.
Then next is the heterozygous.
Wait, so what would the ratio be on the test?
It'll be multiple choice ratios. This is the correct ratio because you've got 1, 2 of these, and 1. So if I asked you to cross big T, little t with big T, little t, which is a heterozygous cross, like you mentioned, using a Punnett square, and I asked you the ratio, you would say 1 to 2 to 1. But on the test, you're not going to have the letters. I just put the letters here so you can see where they're coming from. Yep.
So if they ask a question and it's like cross these two and there's say 0 recessive offspring, right? Would you-
No, you have 1 recessive offspring.
I know, but if the Punnett square was different.
Oh yeah, but they're going to give you what they want. So you'll just plug it in. So in fact, this is the test question. There are no hypotheticals here. All right, so that's pretty easy.
So if there was no little two t's, you would do 1, 3s. Say there was 3.
Yeah, so for example, if you had like, yeah, but you know.
Say you had 2 big t's and then 2 little t's. Would you do 2, 0, 0, and 2, 2, 2?
You never put a zero in.
Never 0.
All right, so next one is phenotype. So once again, the words that I chose are to help you memorize them. So P for phenotype and P for physical appearance. So this is actually what you're seeing now. So phenotype is the physical appearance or expression of the genotype. So once again, sticking back with flower color. If I asked you the phenotype of these flowers right here, you wouldn't give me a combination of letters or alleles. You would tell me the color that you actually see or the physical appearance. So that's the phenotype. So you would tell me yellow, red, there you go, white. And then obviously if you looked at it, it looks like, well, I wouldn't use this as an example. It looks like red might be dominant, but then there's a ton of yellow back there, so I don't know. So phenotype is the physical appearance or the expression of the genotype. Genotype is the combination of alleles. Once again, alleles are represented by letters that you plug into a Punnett square and figure out the probability. So phenotype would be like hair color, eye color, tall, short, seed color, anything that you actually see, skin color. So physical appearance, what you see is represented as phenotype, and the combination of alleles is the genotype. Raise your hand one more time. All right. All right, so just as you had a genotypic ratio, you also have a phenotypic ratio. So phenotypic ratio is going to be what you see. So this could be a test question. What is the phenotypic ratio of a heterozygous cross? So now you know that you have a capital letter with a small letter. Once again, sticking with the same theme and the same Punnett square, which is the first one you drew. The ratio is going to be 3 tall to 1 short because we're looking at physical appearance. And once again, you always do the dominant trait first and the recessive trait last. So you got 1, 2, 3 that are tall and 1 that is short. So the phenotypic ratio and the genotypic ratio, 2 different things. One refers to the alleles and one refers to what you actually see or the physical appearance. Yep.
So the answer for the first question would be number 3?
Yeah, exactly.
Okay.
So, and actually, the handout right here is going to ask you some of the same questions that are on the PowerPoint. So once again, these two kind of go hand in hand. All right, so you should have no problem, you know, after today's lecture of how to do Punnett square and how to plug stuff in and know the difference between a phenotype and a genotype and how to do the different ratios. If you need help, obviously I can help you out. All right, next slide. All right, so a hybrid. So what we've been doing and what you're going to be doing on the test is just this one right here. It's called a monohybrid cross. Mono means one, and a hybrid means one pair of contrasting traits. So once again, we're looking at height, tall versus short. When I think of hybrid, I think of like a hybrid car because it's got 2 different things going on. It's got electricity and it's also got gasoline. So a monohybrid cross is a cross between individuals that involves one pair of contrasting traits. So as you might assume, and this was actually the last question on the book activity, I'd ask you about a dihybrid cross. So di means two, the prefix di is for two, so dihybrid cross is a cross between individuals that involve two pairs of contrasting traits. So in this case, you're looking at two different things going on. So once again, you don't know what the B or the S stands for. They would have to let you know. So you can see that this is a little more complex to do. You go to college and you take a genetics class, you'll be doing that. But in high school, you'll be doing this. So we'll be doing the monohybrid cross. Once again, we've already written this one down. So if you know how to do homozygous cross and heterozygous cross, and those are the only two crosses you can do. And then, of course, you could do a trihybrid cross. So obviously you have more numbers in this, so you're going to have different ratios. Or larger ratios, like 9 to 3 to 3 to 1. All right, so you'll see this on the test too as vocab. It's basically you match a monohybrid cross with one pair of contrasting traits. So once again, the prefix mono means one and di means two. Two sets of contrasting traits versus one, and today this is all we've been focused on. I just wanted you to know that this exists. All right, raise your hand one more time. All right, so the last thing we're going to talk about is the difference between dominant versus recessive, and once again, easy vocabulary. And actually, there is one more thing that we're going to talk about that's probably something you haven't heard of. So hopefully all this stuff is kind of a review for you. So just showing you D1 generation going to, once again, we talked about all this stuff on the last unit, was haploid, gametes, zygote, fertilized egg, diploid. And then just showing you different ratios for a dihybrid cross. And like I said, way more complex than a monohybrid cross. And then reviewing the terminology we've already used. You have the parents, or the P1, then you got the F1, and then you have the F2. Like I said, that's as far as we go in this class, but obviously you go to F3 down here, and you can see it gets more and more complex. All right, so dominant versus recessive traits. Reviewing what Mendel had, you could see that these are dominant versus recessive, so the terminology should be really familiar with you. And so recessive trait is what you're going to write down for vocab. So basically, it's referring to an allele that is masked by the presence of another allele for that same characteristic. So what it means is the allele is there, it's just not being expressed, or it's recessive. And the only time you see the recessive trait is when it's homozygous recessive, meaning you have the two lowercase letters. So the example, once again, using height, capital T equals tall, so they would have to let you know that in the word problem. And then small t would be for short. So if you have two small t's together, that's the only time you see the recessive trait.
Bless you.
Bless you. So if you look at the Punnett square, for example, big T, the little t is still hanging out. It's just not being expressed. So it's considered to be a recessive trait. So it's being masked by the big T. And then when you do it to the F2 generation, that's when you actually see it make its appearance. It becomes homozygous recessive. Raise your hand a few more times. All right. All right, next slide. So recessive trait, once again, referring to an allele as being masked by the presence of another allele. And in this case, you see the big T is masking the little t, and the only time you see the two little t's being expressed is when they're together, and you're going to see that recessive trait. So putting it all together, you've got genotype, two capital letters, homozygous, it's dominant. Once again, this is heterozygous here, but it's still a dominant trait. Two small lowercase letters, once again, this is going to be the genotype is recessive. So a dominant trait basically dominates over all the other traits. So unless it's homozygous recessive. So the dominant trait dominates over recessive traits unless the recessive trait is homozygous, two small t's. So same example again, using height. This is always going to be dominant when it's capitalized. So if you have two big T's, it's going to be tall. Big T, little t, still going to be tall. The only time it's going to be short is when you have a homozygous recessive right there. So dominant, 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 recessive. So once again, these are easy, basic concepts. Like I said, I'm hoping this is a review. And in the packet, I'd like you to do 1 through 14. So you have a couple of Punnett squares in there. There's a little checklist. You check whether it's heterozygous, homozygous, based on looking at the pictures of the alleles.
Are you done with the question?
Now I've got one more thing.
Okay.
Go to slide 60.
Up to number 14. Correct. 1 through 15. I think some of the letters got cut up in the top.
Bless you.
Yeah.
That's all. We can do the different period too.
Raise your hand if you need more time. All right, so last part right here is referring to what is called epigenetics. So this part right here is probably the only new thing that is going into your vocabulary today. So the prefix epi means upon, like your epidermis is the top layer of skin. So a lot of research being done in this, and basically epigenetics may control the expression of our genes. So what that means is there's things that can control your genes that could be like environmental factors, or it could be things like malnourishment, all sorts of things that are out there that could basically have an effect on your genes. That's what epigenetics is. So it involves the modification of the activation of certain genes, but not the basic structure of DNA. So what that's saying is DNA structure is not being changed, but some of the genes may become activated and some may not become activated. So there's two that I would like you to know. And first one is, once again, think about what the words mean. The words are very easy for your vocabulary. So gene is really small. Remember, a gene is a segment of DNA. So if a gene is not expressing itself, it's called gene silencing. So it's silent. So there could be something going on with that gene where it's just not being expressed for whatever reason. Like I said, it could be an environmental factor. It could be things like stress. It could be things like malnourishment. A lot of things could be affecting that gene. The second one, what's bigger than a gene? You basically have a chromosome. So an entire chromosome is not expressing itself, so that's called chromosome inactivation. So these are all new or recent areas of research in genetics. So this stuff wouldn't even be in your textbook. So once again, you've got, as I mentioned several times, you have a lot of DNA, and deciphering what all that DNA does, that's what they're working on right now. All right, so you have gene silencing and you have chromosome inactivation, meaning that an entire chromosome is basically not expressing whatever is encoded within the alleles on that chromosome. And for a gene, which is much smaller, just that little sequence right there is basically not expressing itself either. All right, so what I'd like you to do on your packet is I want you to, not yet, I want you to write your name on the front of the packet, and then I want you to turn to this page right here. So it says, how alike are our alleles? So you're going to fill this in together. Don't jump ahead. So it'll be very, very easy for you to do. You can see what specific genetic traits you have, because that's what we've been talking about today. So I put the example on the board right over here. So this is what it looks like. It's the page in the packet that looks like this page right here. All right, so the way it works is, if you have the trait, you write yes. If you don't have the trait, you write no. And then if it's dominant, that means you probably have the trait, and you can write in the combination of alleles that I'm going to put up on the screen. All right, so the first one. So if you can roll your tongue, you're going to say yes. If you can't roll your tongue, you're going to say no. So if you can roll your tongue, you're going to put the dominant trait, and then you're going to put the genes, 2 big R's, big R, little r. That way you know it's dominant. Yep.
Are you sure that's genetic? Because I used to not be able to do that.
Well, you can actually train yourself to do stuff. For example, if you're left-handed and you want to be right-handed, you can actually train that.
Is it dominant or a recessive trait?
It's dominant. So dominant trait is the ability to roll your tongue.
So then would you write capital R?
What about
Yep.
that?
So big. Calm down, people. Either you can do it or you can't do it. If you can't do it, you can't do it. Right recessive. Next slide. You can ask me when I'm done here.
Oh, gee.
Wait, how do you know if you're homozygous or heterozygous?
You don't.
Well, what if one of them can do it and I just can't?
Okay, people. All right, so next one right here is, you don't have to do this here, you can do this in privacy of your own home, is dominant trait. So if your second toe is longer than your big toe, that's dominant, so you would write yes, and then you'd write 2 big B's or a big B, little b. And then if you don't have a bigger toe, then you would write no, and you'd write 2 smaller B's. So this part right here is showing you, I've got pictures, because that's what I do. That's dominant. And then a lot of what ifs, people, either you have it or you don't. All right, this one right here has a toe ring, so I want you to bring this back into fashion here at school. In fact, make a club, toe ring club. You can have it in my room. Actually, don't have it in my room. Have it in another teacher's room. Why not? All right, next one right here is called widow's peak. This is a dominant trait. The hairline has a point. So, do you have it? Obviously, I don't have it because I'm losing my hair. All right, next one right here is, you look at the picture.
Was it RR?
Yep.
Or okay. Attached earlobe.
Okay, wait.
It means it's like Nikki. Nikki has.
All right, people, I know this is really confusing. Either you have it or you don't.
Wait.
I got too excited.
Do I?
All right, so
No, but
dominant
Nikki
trait,
does have.
earlobes
I mean,
hanging
that's a
freely from the ear. And then right here, they are attached. So maybe look at your neighbor.
Who has it?
Huh.
I know Alex.
All right, so if you have it, if they dangle freely, it's a dominant trait. All right, so for the next slide right here, this one's going to help some of you out in the future. This is called hitchhiker's thumb. You wait.
I don't have it. Oh,
You can
wait.
make it whatever you want. I made up the letters.
Yeah.
So for example, this one's going to be H.
Mine's.
All right, so
I don't even.
So for example, this right here
How do you do
is
that?
hitchhiker's thumb. If it bends back, I don't know what's going on, but that's accepted.
Is this?
Oh, you have?
I'm dumb.
Wait, is this it?
You have,
Yes.
never mind.
What is
You're
this?
an anomaly. No.
Yeah, it's dominant, have it. But hitchhiker's thumb is this.
Alright, so next one are people like this, hairy knuckles. So if you have no hair on your knuckles, you might have a little bit. And this is actually a lot, so it's actually a recessive trait.
That's Robin Williams?
That is Robin Williams, and that's the hairy knuckles.
Of course.
So no hair is dominant, and if you have a bunch of hair, it's recessive. And once again, the knuckles are located right there.
I have dimples.
Yeah. Alright, this is from Lost. So, dimples present. No dimples, two small letters.
Do I?
Do I?
I like sometimes.
Yeah, you do.
It doesn't. Flat.
Stop it.
Wait, go back, go back.
Wow.